Good evening, ladies and gentlemen. We are very grateful that you came to the 18th Landsteiner Lecture by Tony Wyss-Coray. And I would like to start by acknowledging the players and Anne Harvey Nagel on first violin, Hu Xing Yo second violin, Kurt Hintermeyer at the viola, and Ilyana Tumancheva at the cello. Thank you very much. They will also play two more pieces, one after my introduction to the lecture series, and before the introduction and the lecture to the speaker. Let me start by saying, as you have seen, we have chosen Strauss because it's the 200th anniversary of his birth, and this particular lecture hall, the festive hall of the Austrian Academy of Sciences, is a place that is very close to music, as both Haydn and Beethoven have directed here, and we've had some marvelous concerts also in the 200 years after. Let me start by welcoming you all, and I call this the gathering of all our friends, and we are fantastically happy that we made it to the last seat. So it's the prestige of our speaker that did that. But I would like to mention a few of you, just particularly because we are particularly happy that you have come. Her Excellency, the Ambassador to Switzerland, Simon Mayer. Also the representative of the Medical University, our sister organization, our most important partner in the form of Vice Rector for Research, Michaela Fritz. I would like to also, a person that is particularly dear to this community, the founding member and former president of the European Research Council, Helga Novotny. And I would also like, as a representative of our closest partners, the IMBA director, Eli Tanaka. Welcome, Eli. And all of you. A special mention, actually, to Professor Misha Freismuth, who I think is now attending the 17 out of 18 Landsteiner Lecture, and we're very happy about this, but so did Meinrad Buslinger and a few others. We are very glad you came. So, very briefly, a few things about Sam and about this lecture. So, I know that most of you know us, and I am, let's say, saying this for the few of you who don't, but it's too nice an occasion not to try to make a little bit of publicity for the center, because it is actually a jewel. And very briefly, we belong to the Academy of Sciences, and the highest representative tonight here is Huberto Schmidt-Felden, who I'd like to welcome particularly, also thanking him for the hospitality. We are very happy that we can be here. We are, in fact, a GmbH. We're incorporated, which gives a few administrative advantages, and the co-CEO, Anita Ender, is here tonight. We are roughly 20 groups. I say roughly because it's always a balance. Some are in the building, and the others are distributed around the campus, hosted by the Medical University and other places. We have 70 PhD students, and 300 people in total, which is quite a high number. Despite the fact that this is the 18th Landsteiner lecture that I introduced, I'm still very emotional about this because it's such an important event for us. Most importantly is that we are still a young institution because of this student and postdoc, so it's about 30 years of age on average, and 50 nations, so we are very international, which we are particularly proud and happy about. What do? We do what we call foundational research, so it's what you can call also basic research about the mechanisms of disease, and there are many things that interest us, and we have no real limits, but mostly we are interested in metabolism, in cancer, in aging, but also in rare disorders, and many more things, and we use a lot of different technologies that I won't have much time. And we are full of art, and Peter Kogler did this fantastic facade that you see here, and Peter is here with us tonight, which is a particular pleasure for me. Now, we have made 6 spin-off companies so far, and they all together are employing quite a lot of hundreds of people. And we also are spinning off other institutes, and ITERA, which is an AI in life science institute from the Academy of Sciences, just born a few months ago with Boehringer Ingelheim Foundation, and we're very happy about this. Used a little bit CeMM as a model, and 2 of my dearest collaborators, Anita Ender and Georg Winter, are both directors on top of Michael Braunstein. Also inspired by this is a new center in Palermo, which I think can also be called a spin-off. Now, there is a person up here that you see. This picture was taken this morning, and this person is our speaker of today, Tony Wyss-Coray. And you see how splendid Vienna can be, from Strauss to Peter Kogler, full of art and inspiration. So these are the 5 principles that we follow, and I'll be very brief, I promise. One is we create technologies. We are believing that without the technology you cannot see. You need the microscope to see something. You need the new tools in order to be able to do something. Georges, why don't you sit on my spot? I welcome Georges Brasseur, who was the previous president of our branch of the Academy. The second is, as already mentioned, we are interested about the roots of diseases, so we try to understand what exactly is the origin of a disease. We are, because of our privileged campus situation with the medical university, we're trying to push for medical application of what we are doing. This is not incompatible with doing foundational research. We think that actually this goes hand in hand. And this comes from this privileged situation. This is a very old picture of the fantastically dynamic medical university campus, Allgemeine Krankenhaus, General Hospital, because in fact, if you would take aerial pictures today, you will see a lot of changes as the medical university is making new buildings. We are in the middle of it. So we are in the middle of a campus that is one of the largest research campuses in the world for medicine, with a lot of medical doctors, 2,000 medical doctors, with a lot of medical specialties, and we are in the middle of it, and many of us, or few of us, half of us, more or less, also have an affiliation with the medical university. It makes it very easy to be dialoguing with the clinical departments, so this is what CeMM lives off. We also have just released our latest research report, and there are many copies waiting for you outside in case you have not received it yet, and we hope that you will take it. Now, very briefly about the Landsteiner Lecture. It started in 2007, so quite some time ago, and it's dedicated to persons or to topics that have really had an impact on medicine but come from hardcore research. We are trying to get people just before they get the Nobel Prizes, because we are not sort of taking, let's say, already hyper-celebrated, although we're always at the border, because certainly the speaker of tonight is already very celebrated. We are so focusing on still active researchers so that they can also accompany us in the years that follow. And we have this amazing hall that we can do this at. Who was Karl Landsteiner? I imagine that most of you will know. He was on the 1,000 shilling note until the euro came up. He was a very important Nobel Prize winner in 1930 for discovery of blood types, something that saved hundreds of thousands of lives following that, especially in transfusion during the war. But he did much more than this, and I don't have much time to go through this, except a political statement that is that even somebody as accomplished and as important as he, but he was not quite that well known, had to leave Vienna in the early 20s. Vienna had been, until the beginning of the First World War, a beacon for medical research all over the world. Americans would actually come to learn and study medicine from New York to Vienna. The war and other things changed it, but also anti-Semitism created an atmosphere that people like Karl Landsteiner thought they would better go somewhere else before it gets worse. Of course, it was also true that the economical circumstances were not very favorable. We have lost a lot of people. He went on to Rockefeller, and most of his major discoveries he did in the Netherlands and in the United States. We have had amazing speakers. We are very lucky. We seem to convince this is how it goes, right? If you start having a few very good names, then other good names follow. I'm not going to go through all of them, but as I say, some of you have been true followers of this series, and I invite you to always think about the month of May as also the month of the Landsteiner Lecture of CeMM, so that in future you will put it in your calendar. Okay, and now we are, before I introduce Tony, I'd like you to listen to the polka. Now, if you are, let's say, normal-blooded, you will have an impulse to dance. It's not impossible to dance here. There is some space. I'm not sure that... so if you feel like it, and some young people would say, you know, I danced at Tony Wyss-Coray's lecture, do that. I may be tempted to take my wife and show you how it works, but I'm too much of a Rampensau to actually do it. But maybe some of you, some of the younger people, Christoph Binder, a true Viennese, maybe can show us how it's done. I shut up. Thank you. Dankeschön. Okay, and now we come to the main course. That is, of course, our 18th Landsteiner Lecture, Tony Wyss-Coray. Tony is originally from Switzerland. He came here with his charming wife, Christina, whom I forgot to say, both of you I forgot to greet, but I do it now. We are delighted that you both came. We hope that, I know it's a bit late in your beautiful development, but we hope that you will remain in love with Europe so that we will see you and get access to your, let's say, wisdom for many, many years to come. Tony is the D.H. Chen Distinguished Professor of Neurology and Neurological Sciences and director of the Phil and Penny Knight Initiative for Brain Resilience of Stanford University. Now, I would like some of the wealthy persons in this room to savor the beauty of investing money to be able to make an initiative that is dedicated to brain resilience. How much more interesting is it that all the art that we are supportive, or all the music is supportive, where would we be with our brain? So the ultimate art project is to support brain resilience. I say this because it is really through the work of Tony that this concept of being able to do something to the brain, biologically do something to the brain, actually became a reality, something that maybe had been science fiction before. As I said, born and trained in Switzerland, he studied in Bern microbiology, where he got his master in '89, and then he started working on immunology in Bern, where he actually figured out that T cells were also antigen-presenting capability, and he wanted, and he told me that it's still not quite understood why T cells would be able to do that. Usually, they are the opposite. They are the recipient of cells that have that property. So from early days, he'd been focusing on intriguing questions. He then did a postdoc at Scripps, a very important postdoc, where he started to do the kind of research that he then continued doing, which is to study the relationship between inflammation and brain capabilities. As you know, inflammation, Entzündung, is the fundamental process by which our body is able to call for alarm to take and check whether something is wrong. And it is a vital and very important process that often goes a little bit overboard and creates problems. Now, with some very clever combination of genetics and immunology, he was already then able to show that you can create situations where you can monitor the effect of changes in brain cells in order to attract or not the immune system. He moved to Stanford, and he's been at Stanford, he's been at Stanford ever since. And he has been doing a fantastic career there, being a person that has tackled this neuroinflammation problem with many different tools. I think that after getting to know him a little bit better, it became clear to me that what made him so successful in his quest was both being fearless and not being shy of doing things that he had not done before, going past the discipline, so trespassing into other disciplines, so having a multidisciplinary approach to the problem, and asking, of course, the important question, which is, what is it that actually ends up in the brain, and what is it that actually is causing changes in the brain? What is the physiological dialogue between the brain and the rest of the body? Is it governed by inflammation? Is it governed by metabolism? Is it governed by other things? And I will not preempt his amazing talk. Sufficient to say that he is a person that mostly has done to understand how the dialogue that I was describing to you can actually help at least offset some of the cognitive problems that we are seeing in neurodegeneration, in aging, and so on and so forth. And the last 5 years have been nothing less than spectacular. I didn't go past that, where in the last 5 years he had 15 papers in Nature. Now if you think, bibliometry is boring and, why is this guy still talking? I'm a baby boomer, so I still talk about bibliometry. But the most important part is that everybody in the room here appreciate what 15 papers in Nature in 5 years actually mean in terms of concentration and intensity of the innovative impact that you're having. And most importantly, one of the things that he has done in all the studies were trying to understand how do molecules actually go there, what retains them, is there an exchange, do they come out, was the finding that you could create some sort of halting or even improving of certain parameters if you were allowing certain plasma factors in the blood, in this case of mice, to penetrate an aging mice and then help that mice with their brain. What you could call some sort of the molecular basis for Dracula or something like this. So, ladies and gentlemen, we are delighted that Tony has accepted our invitation. We hope that he will remain a friend of CeMM and a friend of the Academy of Sciences and a friend of this community. And without much more, I would like to ask him to come on stage and give the 18th Landsteiner Lecture. Thank you for coming, Tony.
Thank you so much, Giulio, for this wonderful introduction. Thank you all for having me. I just had an absolutely amazing weekend. Stephanie and Giulio treated us, Christina and me, to the opera, to the Musikverein, and it was really absolutely amazing. So thank you so much, also for the organization. It's been really fantastic. So I have used this title, Young Blood for Old Brains, a number of times, and I want to give you sort of an overview of why I use this title, but also, tell you a bit about how we arrived there and give you an overview of the type of research that we have been doing, but also hopefully share some of the excitement where this could be going. So I want to talk about the human brain. The human brain is just this absolutely amazing, the highest achievement of evolution, if you will, or of God's creation, whatever position you take, but it's just this amazing supercomputer that is based on a genetic blueprint. It contains billions of units, we call them neurons, and trillions of connections between them. We call them synapses. And that allows it to, over many, many years of development, I wrote here, the peak performance is achieved after 9 months of construction and after about 25 years of learning. So it takes a very long time for it to achieve the peak, at least as we measure sort of from when the maximal amount of sort of structural functions that we can measure occurs. And it also, of course, the seed and the origin of self-awareness, of socializing, of communication, it makes us humans, right? And that's why it's so devastating if we lose that capacity. And unfortunately, we do. Even the young people here, they don't think about aging. Certainly when I was starting my career, I never thought about aging. That was the last subject I wanted to study, to be honest. But you see here, this is a graph that just shows from left to right, people aged 20 to 90, and it dissociates sort of cognitive function into different domains of vocabulary, processing speed, spatial visualization, memory, and reasoning. And apart from getting more sophisticated in your language, so your vocabulary keeps increasing as you get older. You know, more and more SAT words, as we call them in the US. All the other stuff goes down. And many of us, we just talked earlier how often at social events, how terrible and annoying it can be if you forget a person's name, right? And again, when you're 40, that doesn't happen to you, or very rare. But when you're 60, when you're my age, it occurs more and more, and it goes down. You see where this is heading. So our brains, as we get older, as most of our body, but the brain in particular, because it makes us human, it becomes impaired, and it often leads to neurodegeneration and Alzheimer's disease. And so to just emphasize this a bit more, at age 65 to 70, roughly 3% of people are severely impaired cognitively and demented. That increases dramatically in people above age 85. Roughly a third have dementia. And then in people who are lucky enough to live to 100 years, maybe up to 70% are either impaired or demented. So dramatic numbers, and it's really something that affects us more and more because we all get older as a society, not just as a person, but there's more and more older people because we figured out how to survive heart disease, of course, infectious diseases, but increasingly also cancer. So people live longer across the globe. So every society, as they mature, has these problems. But I also want to emphasize here the positive aspects, and this is resilience, so the bottle half full. What you actually find is 30% of centenarians have their cognition intact. So what does that mean? It means our blueprint, the DNA that we're given, has the potential to reach 100%, not in all of us, but in some of us. So that means there is the potential to figure out how this is achieved, how this is achieved in some of us, what we call resilience to age, and potentially develop medicines out of that knowledge. So then why am I here talking at the Landsteiner Lecture, who discovered blood as an organ that is different between people, a tissue that is different between people. There's these different blood groups. And that really led to the first blood test that we used in the clinic, and we still use now. If you transplant the tissue, you need to know what Landsteiner discovered. So what we try to do is to actually study the blood, as you see throughout my lecture, and to learn more and more about it, and really build on that fundamental knowledge of what is in the blood, how does it make us different, and how can we learn from that and benefit society and humanity. So blood is really a magic fluid. It is used to monitor health and disease on a routine basis. And it is also, and Giulio briefly mentioned this, he mentioned Dracula, but this is actually another rendition of sort of this magic idea of the fountain of youth, that something in blood, in young blood, may have the capacity to keep an organism young or make it even young again. And so transfusion of young blood can rejuvenate old brains, is what we were able to show, and many other organs. And we were not the only ones. There were many other people who started to reproduce these findings, but also show them in different organs. And then lastly, blood can be used to study brain age and function, as you will see. And I think, again, this has potential to get much broader and potentially have impact on a regular basis on medical care. So again, coming back to this concept of what can you do with a drop of blood? When you go to the general practitioner, they usually take a blood sample and they measure a number of different things. They measure electrolytes, they measure a few proteins and other metabolites that tell them whether your kidney is working well, whether your liver is okay, whether your heart is working well. And this is currently only a few dozen analytes that are being measured, maybe 100 tops. But now we have the tools, we have molecular tools to measure tens of thousands of different molecules. And this is really what we have been taking advantage of, as you will see. So the blood is this tissue that is at the center of organismal function in our bodies. It connects all the different organs, and it thus also collects information from all the organs. It serves communication between tissues, and it, of course, also transports cells, like red blood cells or white blood cells, the immune cells in our body. And so when you look in a drop of blood, you find proteins, you find molecules that come from all kinds of different cells in our body, including potentially from the brain. This was sort of not appreciated 20 years ago when we first started doing that. In fact, most neurologists who study the brain, they never collected a blood sample from a person because it was conceived that the blood is sort of separate from the brain, or the brain is an isolated organ. But when you look in the blood in general, what we now know is almost every protein, every molecule that is produced in our body is ending up in the blood or is in the blood if you have sensitive enough measures. And I just show here in the middle, some of these examples of molecules that you might find in blood. We don't have to go into details, but as I said, tens of thousands of different ones. For convenience, because we know how to study them, how to manipulate them, we focused on proteins, but not because we think other molecules are not important, like lipids or sugars or other types of metabolites, for example. So we focused on proteins, and also because proteins can now be measured more and more easily. And as I said before, so there are proteins that could come from the brain, but they could also come from any other tissue that allows you then potentially to get insight into the function, and as we will see, into the age of that tissue. So could we then take a sample of blood and study how it changes with age in the population and learn about the effects of genes that make us different, about environment, what we eat, lifestyle, and so forth, and learn, based on the changes that we can observe in the blood, about biology and about risk for disease. And so here I show you an example of a study that we did a few years ago now. This was not a study we initiated or conducted, but we obtained samples from this study. It was a healthy aging study with roughly 4,000 people, age 18 to 95, so the adult population. They were all considered healthy, so they didn't have major diseases. And again, we obtained data from blood samples from these people for 3,000 different proteins. So these proteins may have very different functions. It's a sort of a collection of different proteins that were put on this platform. And so when you look now at a protein across lifespan of people, so we have actually these 4,000 people, and if you look at a protein such as this one here as an example, it may not change with age at all, right? It may be the same level across the population, whether you're young or old. And we use two colors, blue and yellow, to sort of express the change or the levels of a protein. So if it's low throughout life, we show it's blue. You may think of a protein that is low when you're young. It goes up with age, an inflammatory protein, for example. Or the opposite, a protein that is high goes down with age. Some growth factors show this behavior. Or you may find a protein that goes up and then it goes down again. And you literally find any type of trajectory, as we call it, with age in these proteins. So if we look at all these individuals, 4,300 people from left to right, and then for each person we measure 3,000 different proteins. And we generate this Roy Lichtenstein type painting, where you see these beautiful changes that occur with age. What you immediately notice that from left to right, there are dramatic changes. So it seems like no sample is the same as the other. And this is only one sample per person, but again, from 4,300 people. You also notice that these changes are not linear. So when I showed you this protein that just goes up, there are such proteins, but the changes overall are not what we call in a very predictable fashion. And in fact, it goes, sort of slightly random, but if you actually, if you squeeze your eyes a little bit, you notice that there's sort of major changes happening. We can call them inflection points. And the first one, if you look at this, so if you count all the proteins when they change with age, the first one you see is sort of here around age 30 or 40. So again, young people, don't think you're not aging. There is something that is happening at that age. We call this waves of aging. And we describe them here for the first time at scale in a human population. And since then, other people have shown, actually with different types of molecules, that you see this wave-like behavior. We see a slight wave at age 65, and then a dramatic change at age 80 or so. And again, this just reflects changes in protein levels. So there are massive changes in the concentrations of many, many proteins at around age 30 to 40 in people, and then another one 65, and then the biggest one at age 80. And certainly the last one coincides sort of with the frailty that makes people incredibly susceptible to age-related diseases and death. The first one is sort of curious, right? And we speculated, entirely speculative, that this could be associated with sort of the evolutionary age of humans. So without any medical intervention, hygiene, or anything, humans lived on average, 30 to 40 years. That's enough time to reproduce and guarantee your offspring. And so maybe nature didn't care about us anymore. And so there are just disappearance of maintenance mechanisms that are necessary to guarantee the species continuation. And once that's guaranteed, nature unfortunately doesn't care anymore, and you have really sort of the first wave of age-related diseases coming up. Again, speculative, but hopefully we will find more data in the future. When you look at, I showed you some examples of trajectories of proteins. So what you can do, you can use algorithms that pick out proteins that have similar changes. So let's say proteins that all go up with age, or they go down and come up again. And so if we do that, we call this clustering. We basically look among these 3,000, and this is actually a different study here. We have 22,000 individuals with 7,000 protein measurements, and we get very similar results with the previous study. When we pick out these clusters, what we see is that there are groups of proteins that show the same behavior, and interestingly, they often have shared biological function, or they originate from a tissue or from a cell. So what do I mean with that? So if we look, for example, at this orange cluster here, cluster 12, that goes down with age and then comes up exponentially, it seems, in very old people, maybe age 70. If we look at this cluster 12, we see that it's a cluster that is associated with immune cells. So it contains proteins of the immune system, hundreds of proteins, 200 proteins, and the majority of them seem to be from the immune system that show this behavior. There's another one here, cluster 33. This one is looks in the gut-associated cells. And then this one here, cluster 26, which goes up, stays flat a bit, and then goes up again. That one seems to be associated with the brain. So it contains a lot of proteins that are derived from the brain. And you will see why I show you that, because what we can do with this knowledge is we can learn something about the proteins or the tissues, or the cells or the tissues where these proteins come from. So we can then ask, are these changes that we see, are they the cause of aging, or are they just a result of aging, right? So it could be that these changes that you see are just a reflection of the aging process. But it's also possible that some of these proteins, they actually cause aging, or they try to stop aging, so that they have a functional effect in the aging process. And to figure this out, this is where Dracula comes in. So if you think about, if we have young blood and old blood, and they're completely different, so would it be possible, crazy idea, that you take the blood from a young organism and you put it into the old? Would that change that picture that I showed you with this yellow and blue colors? Would that make an old person look younger again? I don't have the result in people, but it works in mice. And when we initially did these studies, they seemed too good to be true, right? And there's too much mythology about young blood and Dracula and all of that, that it seems this must be crazy. But it does indeed work. And I was not the first to do this experiment. It was actually my mentor, Tom Rando, who asked a very fundamental question. So he studied aging of the muscle. And in the muscle, there are cells, they're called stem cells, that keep producing new muscle tissue. So if you go to the gym, you work out hard, you make new muscle tissue. If you get older, you actually don't make muscle tissue anymore. Sorry. You make scar tissue often. So hard workout can lead to scarring of the muscle. And it's because these stem cells fail to make muscle tissue. So Tom asked a very similar question that we had. What if he changed the environment of the old muscle stem cell and exposed it to young blood again? Could he find out whether there's a problem with the cell itself? We call this intrinsic. Or is it something from the environment, from the blood that is missing with age? And I wouldn't be standing here if he didn't find that young blood can actually rejuvenate these old muscle stem cells. And they actually, histologically, they look almost like young muscle cells and like young muscle tissue. And he found that there are potentially effects on the brain as well. And that's where we started to collaborate, because again, we had this observation that the composition of the blood changes with age in mice, but also in people. So what if we exchange that? Would we have an effect on the brain? And indeed, what we found is that the brain also has stem cells. We could reactivate them, rejuvenate them. And we saw that they made more mature neurons. We see more of these synapses talking to each other again in the brain of the mouse. We saw less inflammation. And most importantly, mice, they have actually, when they get older, they have a similar problem. They can't find spatial locations anymore, a problem that many Alzheimer's patients have, that they can't find their way home. So we can model that in mice. And when we gave them young blood, they could do this test much better again. And there's multiple different tests that we can use, and they all showed similar results. And then other people had similar findings in many other tissues. And now it's pretty much accepted that there's true biology underlying this effect. And we can actually take now the cells from every single organ in the mice and show that they respond to this effect. What we also found, there is this cerebrospinal fluid. That's the fluid that bases our brain. If we collect this from young mice and infuse it into old mice, into the brain, we can again regenerate and rejuvenate these brains, and they function like younger brains, and then also reverse many of these age-related changes that occur in the brain. But also what's very intriguing and baffling in a way, if you do the opposite, if you take blood from an old mouse, you put it into a young, you can actually accelerate aging. And again, at a molecular, cellular, and functional level. And we can show that with many different tools and with, in many different cell types. So it's not that everything would be going from the right side of this figure that I showed you to the left side again. But there are clear indications that some cells are very susceptible to rejuvenation, and that leads to a functional recovery of the tissues and the organs. So at this point, we cannot make an old mice get smaller and look like a young mouse again, like Benjamin Button. That doesn't happen. But we can rewire and make many cells and genes and molecules look like those from a younger organism. This is actually a very convincing experiment. That's from a good colleague of mine, Vadim Gladyshev, and his fellow James White. They used a model. This is a bit gross, so you can close your eyes. But basically, they suture animals together at their flank. It's called parabiosis. And that leads to joining of the blood system in these mice. This was invented by Paul Baer over 100 years ago. It led to discovery of tissue rejection that we use now for transplantation, sex hormones, or leptin, which is this obesity hormone. But here it was used to study whether the blood from one animal influences age of the other animal. So they made pairs of young and young mice together, old and old mice, and then the young with old. We call this heterochronic because they have different ages. They left them together for 3 months and then surgically separated them again so the mouse can run around, and then left them age and asked, does a mouse, an old mouse that was paired with a young mouse for 3 months, does that live longer? And it does. So they show that these old mice that were paired with a young mouse show a lifespan extension on average of up to 10 weeks. So that proves that, first of all, again, these physiological effects, and they're not just limited to the brain, but they are affecting even the lifespan, which is a very complex interaction of all the different organs and tissue, that can be influenced here. Now, as a caveat, these mice underwent 2 major surgeries, right? So first suturing them together, then taking them apart. But not few of us will undergo major surgeries, and it's a key risk factor for dementia when you're older. What they could also show is there are these called epigenetic clocks where you can estimate the age of a cell, of a tissue, of an organism based on changes that happen in the DNA. And so they use this approach to prove, actually, for many tissues that if an old tissue, like the liver here, is exposed to young blood for 3 months, it shows an epigenetically younger age. So again, a molecular evidence that somehow the clock is turned back and it's rejuvenated. So just think about this concept for a moment, that there is a possibility that factors, in this case, in the blood, can slow down or reverse aging and improve function. Now, I'm not telling you should go and perfuse yourself now or get a surgery with somebody younger, your age, but it tells us that conceptually it's possible to slow down aging or even reverse aging. And there's actually many other examples beyond young plasma that can do this now. So it's a reality that we can manipulate aging in living organisms, and maybe even in humans. So what we try to do is to use a paradigm that is done in the clinic in hundreds of thousands or millions of people every year, blood transfusions. And so we worked together we started a company called Alcas and worked with Grifols, which is one of the major producers of clinical products that are made from plasma. So plasma is the yellow liquid that comes from blood. And so if you are immunodeficient because you survived a cancer or you have an autoimmune disease, everybody is on treatment with antibodies that are collected from these plasma donations. So here what we did is we recruited volunteers into a study, Alzheimer's disease patients, into a study where we gave them infusions of plasma from these overall young donors. And we could show in these small proof of concept studies that this was safe and that it looked promising. Now, small studies can never convince that it will work at scale, right? So these were so-called phase 1 or phase 2 studies. But Grifols actually ran separately a study where they removed blood first, which is called plasmapheresis. And then they gave young plasma. So they did a combination where you first take out the old blood, and again, that's an approved technique that is used in the clinic all the time for people with sepsis, for example, or with autoimmune diseases where you want to remove bad factors in the circulation, and you almost wash the blood, if you will, and then they gave them plasma from young donors. And they did this in actually a placebo-controlled trial with with 350 individuals with Alzheimer's disease, and they could show that there were significant improvements. This was a phase 2b, phase 3 study, but it was not a full-size phase 3. There were different doses. So a phase 3 still has to be done, but people have more and more evidence that there are actually molecular changes that are consistent with a beneficial effect on multiple different tissues and organs. In Texas, there are clinics that you can just pay if you have money, and you can get a blood transfusion. But again, it's not a clinically proven FDA-approved protocol. And personally, I think, first of all, even if this worked and it was the magic bullet, we would not have enough plasma to treat millions and millions of people. But for me, it's the proof of concept because it suggests that there must be something in there that we can potentially isolate and turn into a drug, if that makes sense. So I want to show you now more what we can do with this type of information. So imagine we had a sample from the person there in the red square. You can easily imagine if I take this sample, it's like a puzzle. It will fit only at one point of this whole scale of 100 years, right? What does that mean? It means that you can estimate the age of a person. So if any one of you gives me a sample, a blood sample, I can tell you pretty much how old you are. And it's not so magic, right? We do this all the time. We look at people's faces and estimate how old they are. This is a machine learning model that is built into our brains based on experience. People tell you how old they are, and you store that information. And then if you have a new sample, a new person, you make that estimate. You're not always right. And that's actually the basis of even more interesting information. We call this the age gap. So the difference between the person's age and what you have estimated turns out to have very important biological information. So we can use machine learning to estimate how old that sample is based on how similar it looks to the appropriate age. We estimate this person is 35 years old, but as I already mentioned, what this model does, it provides us biological information because it turns out you don't need 3,000 proteins to make that estimate. You need only a few. And the ones that you use, they must have something to do with aging, right? Otherwise, it wouldn't work. You couldn't tell how old a person is if it had nothing to do with aging. We don't know whether they cause or respond to age, but it must have something to do with age. And so is it then possible, using this idea, that you could estimate how old a person's brain is? So imagine you have a blood sample from a person, and some of the proteins that you can measure come from the brain of that person. And now if they change with age, you should be able to tell how old they are. And so how old is your brain? Do you have a brain disease? Could we measure that? I mean, that's not what we typically do, although the field is now moving into that direction. We can measure the pathologies of Alzheimer's disease now in the blood. This was inconceivable 20 years ago. And then, are you at risk of a disease? So what if your brain would be estimated to be older than you are? Are you then at risk for a disease of the brain? So again, to reemphasize this again, so if we look in this drop of blood and we look at all the different proteins, we can ask for each protein, where does it come from? Is it produced only in the brain? Then we can use it to achieve information from the brain. Many proteins are produced throughout the body, so we can't really learn much about an individual tissue or an individual cell. But let's say these orange proteins here, they're only made in the brain, and we can measure them in the blood. And then we can build these machine learning models where on the x-axis, so each dot is a person. Here is what we call chronological age, your birth age. So the exact age is time you have spent on this planet. And then the machine learning model, based on thousands of people, hundreds of people or thousands of people, it builds this model. Again, like we estimate a person's age by looking at the face, because we learned this from looking at hundreds of people, and they tell us how old we are, how old they are. You can use the same model. And then, as I mentioned, the interesting thing is if somebody is not matching their actual age, but they're a little bit older or a little bit younger than what you would predict. We call this the gap, the age gap. And what you find if you look at a population, if we look at all of you, some of you will be exactly matching the age that you are, the chronological age, and others will be a little bit off, and some will be off a lot. And so we get this mostly normal distribution, we call it. But it's interesting to look at the extremes, and we will do that again in a minute. What's really fascinating and incredibly exciting in this field is that, as I mentioned before, the age gap, so the difference between your actual age and the predicted age, contains information about biology, about disease risk, and even mortality, how soon you're going to die or not. And I'm taking a bit of a mouthful here because we know this only from studying maybe 80,000 people now, but it's a reasonable size, and it reproduces across different cohorts in different labs now. So just another example, if the heart, it's blue here, unfortunately, you have heart proteins, 2 heart proteins. They're currently measured in the clinic. If you go to the doctor, they measure 2 of these proteins to tell you your heart is okay or not. Again, the same thing, you can make a model, predict the age of the heart. So I had 2 graduate students, Hamilton O and Jared Rutledge, who did this and described these organ clocks, you may call them. And here we see an example of what we call a heart ager. So this person has a heart that is sort of at the edges of this distribution, has a really older heart than most other people. And we find maybe about 2 or 3% of the population who are extreme organ agers above age 50. So this is from 5,500 people where we measured 5,000 proteins. And these people here, or at this example in particular, the heart is older, but the other organs are actually not older, or just a little. We show from white to dark red. You see most other organs are fine. So it's not enough if somebody tells you you are older than you should be. You should ask what you mean. Is my hair older, my hair follicles, or my face, or my kidney, my liver? Makes a big difference, right? So it doesn't make sense to give an organ overall age estimate. Although that could relate to your overall organ physiology, but you really would like to know which cell in your body is older or which organ is older. And so here is an example where we look at people 15 years into the future. So we got their blood sample at time 0, and then that's when the blood sample was collected. We predicted, or we measured their estimated heart age. And when we look in those with, so the middle is here, this dashed line, the median, the average population. When we look at those who have the youngest heart, the bottom quarter with young hearts, they had very low incidence of any heart disease over 15 years. But if they had older hearts, so at the top 25%, we see a roughly 2.9-fold increase risk in developing heart disease. So they had heart failures or atrial fibrillation. And again, we didn't know that. We just looked at some heart proteins. We know change with age, and then we can predict, how old is their heart, or we can, estimate how old is their heart. And based on that, they have the risk to develop heart disease. Here you see in another study, this is with 50,000 people, it's this famous UK biobank, which has 600,000 individuals enrolled. From 50,000 people, an industry consortium measured 3,000 protein. Now this is with a different platform, which sort of validates the approach because we use a different company to measure these proteins. And what we find here as well is that people who have older brains, they get Alzheimer's disease. People with older hearts get atrial fibrillation. People with old kidney, immune, intestine tend to get type 2 diabetes. And this is not 100% predicted, but their risk is strongly increased. So we can then ask, what is the strongest predictor of your risk to get Alzheimer's disease? Currently, we know there's a gene called ApoE, and if you have a variant, there's ApoE2, 3, 4. If you're lucky, you have 2 or 3. If you have a 4, you have 1 copy, your risk is increased by about 4-fold. If you have 2 copies as a woman, it can be increased up to 12-fold or 15-fold, your risk to develop the disease. So that's a genetic risk factor. Here we ask, based on these estimates of age of different organs, which is the strongest predictor of developing Alzheimer's disease, and we find it's the brain. So if your brain is older, that's the strongest predictor of Alzheimer's disease. When we then look only at the extremes, so those who are estimated to have very young brains or very old brains, and that's roughly here about 5% of the population, 2 to 5% of the population, we find people, again, with very young brains, very old brains, so we basically exclude what's in the middle here, and we ask, what is the difference between the risk between these extremes? So if your average risk to develop the disease would be this gray line here in the middle, if you're lucky to have a young brain, the people with the youngest brains in these 50,000 individuals, they have a 80% reduction in risk to develop Alzheimer's disease. 80% reduction. That's phenomenal. That's phenomenal. So it means you have a very small chance of getting this devastating disease. And it's like, the ApoE2 allele in Alzheimer's, in this gene that I mentioned. That's actually a variant that is associated with centenarians and people who are resilient. So this is like that, but it's none of these genes. It's also none of the genes associated with the pathology. It's a true aging signature. So we look at something related to aging, and I'll show you in a minute what we're looking at. And then if you're unlucky to be on the other extreme, so you have very old brain, your risk to the average population is 3.4-fold increased, and the difference between these two is 18-fold. And that's simply an aging signature. And here are the proteins that are used to build this model, if you will. So we take all the proteins that are specifically derived from the brain, we pick those that are the best predictors of brain age. And what turns out, these come from different brain cells, not surprising, not just from one cell type, but from different cell types. And when we then ask, what do they do, or what do we know about the function of these types of proteins, we find they're involved in something related to synapses. So synapses have some of the synapses have this structure around them that looks like a basket almost with holes. And we call these perineuronal nets. And this is a very strong signature here for these perineuronal nets. And it suggests that there's some interesting biology about aging of the synapses, which has long been suspected, but here this is really the top biological signature that seems to predict how old your brain is and how much you're at risk of developing neurodegenerative disease. Now, let's go back to looking at the different organs. And now we ask, what is your risk to die? It's not very useful, admittedly, but it's the strongest validation of a model, right? Because you could always argue, when did the disease start, or is it really this disease or not? But if somebody dies, they're dead, usually. So it's a very good readout to validate these models. If we can predict if somebody dies or not, that means we have picked up some biological information that is relevant. So what is the best predictor of mortality? The brain, surprisingly. And when we looked in the literature, there are some hints from fly studies and from other organisms that the brain may have some information about lifespan. We don't really understand it exactly what it is, but it seems to be a very strong predictor. Even though you don't necessarily die of Alzheimer's disease, right? But there is something that we don't understand yet that seems to be the best predictor of mortality. So if you have an old brain, that's bad. And then you can see here some of the other tissues, lung, intestine, heart, and so forth. If you have multiple organs extremely old, so we look only at the extreme ages, so that little sliver on the very right side. If you have multiple organs older, and there's about 2% in each of these categories in the 50,000 people, your risk to die increases dramatically, and you see in a minute in another illustration. We, of course, were also wondering, are there organs, if you're extremely young, that would extend lifespan or would predispose you to live longer? So the best predictor of longevity is to have a young brain and a young immune system, and together you have a roughly 50% reduction in mortality, predicted mortality over 15 years in the study. So, ideally you would want to look over 50 years or more. And there will be studies like that I think people will soon be able to do. So here are what we call survival curves. So at the top here, 1 means 100% people are alive. And then we look across time here. We have, again, the samples were all collected at time 0, where people had different ages, right? These 50,000 people were between 50 and 70 years old, probably. And then we look how many of them die over this period of time. And every time a person dies, it drops a little bit. And if it was at 0, everybody would have died. So the gray line here is the normal population, the average in the middle of this distribution with the age gaps. And then if we look at people who are extremely young, who have this lucky combination of extreme young brain and immune system, they live the longest. It's not such a big difference, it seems, but it's roughly 50% on average. But then if you have increasing number of organs older, you can see here with 8 organs older, so these are really sick people, 50% have died over like 7 or 8 years. So very strong predictor, and we know again what the proteins are that make up that signature. There's also lifestyle information in these studies. It's not as reliable because it's what we call self-reported. So you ask people, did you smoke? Maybe they're honest. Did you drink a lot? Most people not honest. So, deprivation index is maybe a bit more reliable and so forth. But what we see by and large is that what is already known, people who smoke, they have multiple organs older, shown here in red. People who drink excessive alcohol, deprivation index, so growing up in a bad neighborhood. And then some positive things, vigorous exercise is associated with younger organs, education, eating oily fish, and so forth. Maybe a little bit more reliable is use of certain drugs, especially in postmenopausal women, Premarin. That's something that they would remember, right? And so here we actually see, also consistent with the literature, that multiple organs are younger, the heart slightly older maybe, and large studies seem to confirm that. We also saw some of the or sort of supplements that people take, like glucosamine, which interestingly we already found in the first studies with 5,000 people, We didn't want to publish that. We thought this must be an artifact, 5,000 people. But it showed up in this second study, independent different protein measurements, 50,000 people. So if you want to do something, take-home message. If you want to do something good, exercise. But yeah, glucosamine might be good. And a very similar analysis of this study was done by Vadim Gladyshev again, and he actually concluded, which is a very bold statement, I think, and I'm surprised that the reviewers and editors let him put this in, that the age of an organ is really the predictor of organ-specific disease. I'm not sure that's always true, but this is what the data tells us right now. So if you have an old organ, that organ is likely going to develop a disease. And imagine if that is really true, we could monitor people when they're healthy, and they could potentially do something before that organ actually gets disease. All these people that when we took their blood sample, they were healthy, right? So if we could have told them, your heart is older than it should be, maybe they could have taken available medication to treat heart disease before they have a stroke. But we'll see whether that is going to work. So we also built models for individual cells. So our body is composed of thousands of different cells, if you include brain cells. And we were able to make clocks, we call them, not just for organ, like I showed you before, but for individual cell types. And we managed to do this for almost 50 different cell types. You see examples here of liver cells, of smooth muscle cells, macrophages, oligodendrocytes. So these are cells that put the plastic over the wire that connects neurons, if you will. We call it myelin. And so here, again, we use the same principle. We look for proteins in the blood that are only made by those specific cell types and not by other cell types. And we find, again, these extreme ages. So if we look at the overall population, this was from a study with 7,000 people, 7,000 proteins. And now we measure the age of every cell in these individuals with the machine learning models. And then we look, are there people who have extremely old cells? We find these Schwann cell ages. So Schwann cells are part of our peripheral nervous system, and again, they put the insulation over your nerves. So as people get older, they often have neuropathic pain and things like that. And that is in part the result of degeneration of these nerves, but potentially also of the insulation of the nerve. And so you get abnormal conductance of the nerve, and that causes pain. And so it will be very interesting to figure out whether these people have some sort of pain associated with this extreme Schwann cell age. We have people who have a specific type of neurons very old. They tend to have Alzheimer's disease already. This is actually from a study with people with neurodegenerative diseases and healthy controls. We find people who are aged for a specific cell type in your gut. Maybe they are at risk for some sort of inflammatory bowel disease or something like that. And it's just show you this as example of the potential to combine now disease prediction with looking at the aging of individual cells in living people or individual organs in living people. And here's just an example of a first analysis. This is from the study that I just mentioned earlier. So it's a study of neurodegenerative disease from 23 different centers that was put together. 3,600 have Alzheimer's disease, 830 have ALS or Lou Gehrig's disease. This is a muscle wasting disease. 900 with Parkinson, another type of dementia called frontotemporal dementia, 130, and then mild cognitive impairment. And what's the most striking observation here and signal that you can immediately see is what we see here. So patients with this devastating muscle weakness disease called ALS. So Stephen Hawkins had this disease, but he had a very abnormal form that allowed him to live for several decades with the disease. A lot of people die within 5 years of the diagnosis. And it's not sure what are the fast progressors, what are the slow progressors. There's no good drugs or none at all. And what we find here is that these individuals have strongly accelerated aging of muscle cells and also muscle cells in the heart, which was not previously known. But then I looked in the literature and I found a small study that looked at the heart volume of people with ALS, and they seem to have less muscle tissue. And some, they die suddenly of heart failure. Very rare, but so there seems to be something to what we discovered here in an unbiased fashion. And there's a lot more here that I don't have time to go into. We can also, again, estimate what is the likelihood that somebody with old muscle cells, for example, has ALS. And we get some very large, what are called odds ratios. So it's very likely that there is a connection there between these. We find also interesting links between other cell types and Parkinson's disease or frontotemporal dementia with these horizontal cells that may be similar to other neurons in the brain. So my last piece is sort of a little bit esoteric science. Not esoteric, but hardcore science, maybe is the word. So if you don't understand it, I hope you forgive me, but I just wanted to show you that, so how can we actually know we're sure, right? We say this protein comes from this tissue, from this cell type. How do we know? It's very difficult to do that in humans, but there's a trick that we can use in mice, where, and this is work from Ian Goldner, very talented fellow in the lab. So we basically hijack a machinery that builds proteins in our body. And to make proteins, you need these different amino acids. And there's a very elaborate machinery that pairs an amino acid with a molecule that is called a tRNA synthetase, and that recognizes the code on the genome and then puts the right amino acid in the right place. So this is how we synthesize proteins. And we can hijack that. So we make a small mutation in one of these mutant tRNA synthetases so that it accepts a slightly different amino acid. In this case, we put phenylalanine with an azide tag into the growing protein chain instead of the regular phenylalanine. And the reason why we do this is that's a chemical modification which allows us to use click chemistry, something Caroline Bertozzi, who has worked with us on this, got the Nobel Prize for a couple of years ago. And so basically what we do, what we generate is, this is the word for it, don't try to read it. So what we do is we make proteins that have these little tags. So we almost label them. And what that allows us then is to fish them out and study what are they. But we can put this machinery in specific cell types. So what Ian did, he put this thing into neurons of mice. So only neurons have the machinery to put this azide phenylalanine into the growing protein chain. Essentially what we do is we tag proteins that are made from neurons. So why would we do this? If we now discover any protein that has a tag, we know it must come from the neuron because only neurons have the ability to do this. And so now we can collect brain tissue, and then we see lots of proteins that have it. We can collect the spinal fluid, and we can measure many proteins that come from neurons that have the tag, but we can also do it in blood. And we show for the first time proof that proteins in the blood must come from neurons, and we know which ones. And so this just shows that the method works. So the beauty of the system is also that for the first time you can actually look at neurons, which are very complicated cells. They have a cell body, but then they have these very long processes called axons. and then they have many, many different connections at the end. So it's hard to isolate them from tissue because they always fall apart. But if you label them, so these neurons now have the machinery, and we can see hundreds of or thousands of proteins that cover the entire structure of these neurons. And even if we take neurons from different parts of the brain, they have slightly different, what we call signatures. And now we can ask, as I said before, if we detect a protein that has a tag that has this star on it, it must come from a neuron. So somehow it made it out into the blood, and we can measure it, and so we can prove that this protein comes from neurons. And this is really very new work with Emma Landberg, who is a protein chemist from the Karolinska, and she has one of these top-notch instruments that allow you to measure very, very small quantities of proteins. And we can measure over 600 proteins in the blood of these mice that must come from neurons, so different proteins. And what's really exciting is many of them, roughly 100, come from synapses. So they come from these essential units that neurons use to talk to each other. And we know that the synapses are the ones, remember the signature for brain aging? These are synaptic proteins. We measure some of them that are shown here, and they make up that signature. So while we can't do this in humans, by analogy showing in mice that we can measure all these synaptic proteins that correlate now with function of the brain, we can do all kinds of interesting experiments and hopefully demonstrate that we can measure brain function in the blood indirectly. And with that, I want to summarize that plasma protein changes with age provide insight into global aging patterns, relative age, and the physiological state of cells and organs, so the functional state also of the cells and organs. Protein changes reflect physiological state, but are also drivers of that state, shown by rejuvenating effects of young plasma. So again, it's not just a response to aging, but it also seems to actively modulate aging. Plasma protein organ and cell signatures identify extreme organ agers who are particularly susceptible to organ-specific diseases. And overall, we believe that really monitoring the blood, like Landsteiner started to show us, if we do this at scale and we use the modern technologies that allow us to quantify thousands of different molecules, that this will fundamentally change medicine, and we will hopefully go towards healthcare and away from sick care, what we're currently doing, wait until somebody is sick and goes to the hospital. So with that, I would like to acknowledge my lab, of course, many different collaborators and funding sources, and for your attention. Thank you so much.
Thank you very much. We are all stunned and are thinking about the implications of this. So who wants to ask the first question? I mean, the first question is the genetics of this, right? There must be somebody, because you can correlate at least some, the most important proteins and the origin, and you must be able to do the genetics to some extent.
Yeah, this is actually what I'm most excited about. My wife keeps telling me you get more and more excited about stuff, but this is what scientists' problem is, right? We're always excited about the next thing. So we started to use this idea of extreme phenotypes. So for any protein that you can imagine in the population, there's sort of an average level of a protein, and then there are extreme values. And so each one of us has extreme values for some proteins. And if you look at these extremes, you can use genetics, like in a case control study. And there's incredible exciting new information in there. And so that's where we take this. Of course, you could also do genetics on the age gaps. We have done some of these studies, but it's a bit harder to get the power, I feel. But with these extreme phenotypes, we call them outliers, we're very excited about this, yeah.
Fantastic. Christoph Binder first.
Thank you very much. So I'm a laboratory physician, so I'm very close to what you're doing, of course. And I have one comment is regarding the reliability of plasma proteomics. I've recently seen actually a study where they compared Olink proteomics with another platform, and only 50% of the proteins really correlated. So before we get into clinics, I think we have to take care of the reliability of these measurements. But nevertheless, so it's clear that in laboratory medicine or lab values, there is changes over time. We have different cutoffs in young and old, but there's a notion that is becoming more and more appreciated now that you look at individual changes over time, so individual trajectories. So would you think that if you would also consider these in any of your measurements, that you would get an even more accurate predictive power?
Yeah, thank you so much. And thank you also for your comments on, protein overlap. And so when we talk about the protein, we actually talk about a proteoform. So a given protein can exist in many different flavors. Like if you said, I saw a rose, there's many different roses. So for a given proteins, there are many different forms. And for most of the tests of these proteins, we actually measure different forms of the protein. So it's expected that they do not overlap, but you need to know what you're measuring. That's what the clinical chemistry knows, right? So they know what they're measuring. So that has even actually much more potential because we can measure different isoforms and they contain different information, as cancer research is now showing and demonstrating. But you're absolutely right. Looking at longitudinal trajectories is really where the field is going. So we're trying to get studies where people donated multiple samples, and then we see already some consistency in the estimated ages for a given person, but there is noise in the data. But overall, a person with an aged heart seems to, over several measurements, keep that old heart. But it's very early, it's experimental, and we're not in the clinic yet. Yeah.
Okay, before we move on, so we hope there are going to be a lot of questions. Maybe we can question quite a lot. But Tony gave a fantastic lecture, especially for lay people. So we don't want to have only the professors of medicine asking questions, although we love them and we want them. We also would like to give a chance to you to ask questions. It doesn't matter. There is no such a thing as a stupid question. Tony is particularly interested in the questions that come from people that are not of the field because they're often quite enlightening. I have in list a gentleman in the back. I think maybe if I don't see you, it would be nice if you stand up and tell your name. Then I have Christoph Bock. Then I have Misha Freismuth. I have Nikki Kral. And this is the first wave. I'm sorry, you're not a gentleman, you're a lady.
Thanks. I'm a lady, yes, Julio.
Hello, Jana.
Hello. All right. Yes, thanks a lot for the great talk. I have two questions. On one hand, you were showing that young blood can rejuvenate, let's say, tissue organs in old mice or maybe also in humans. To what extent is there like a perfect age? Is there like 25 the perfect age for such a blood transfusion? Is it better to take younger blood, or does it depend on the organ? Because like there's different clusters that show like maybe a reduction at 35 and then they increase again. So that's the first question. And the second question is, you showed that elderly people, some have, let's say, like protein signature that correlates more to younger brain age. But are there also proteins that like decline no matter what, but then other protein signatures, like other proteins can, let's say, compensate for this, and that's what makes the difference in elder people between those that are healthy and those that are diseased? Thanks.
Yeah, let me try to answer the second question first. I think we don't know enough, but there's definitely, proteins that just very steadily increase with age. GDF15 is an example. And it's possible that one individual has a slightly different change in proteins that determine their protein age, right? It's not that everybody will, let's say we have 20 proteins that determine brain age. There is sort of a different weight for the different factors, and that may differ from one person to another. I'm not sure that answers your question exactly. But to your first question, younger blood seems to be better, although we have not systematically done that. But we used, for one study, we used cord plasma. It's the youngest plasma you can get from human cord blood. And that contains a large amount of growth factors, not surprisingly. And that had a more potent effect in mice than from adult young mice. But we have not done it systematically.
Okay, we have Misha Freismuth next.
So what struck me is you had these environmental factors like poultry, beef, and so on, and tea. What struck me on your list was that coffee didn't pop up. Because you know that there is all these papers that show that coffee consumption reduces all-cause mortality. So do you have an explanation why you didn't find coffee?
It's a British study. It's very simple. But thanks for pointing that out. It actually never occurred to me. And tea is on there, right? So tea is good. But no, I don't know. But, it is a British study. So, people don't drink as much coffee there. But, as I said, take this with a grain of salt because it's self-reported. And, there's thousands of studies out there that have been done in a clinically controlled fashion. Many of them in academia, and they have blood samples. So there is the potential to now measure these samples and see, do you have an effect with this or that? Yeah.
Christoph.
Thank you for the great talk. It seems that what's really holding back the field of human anti-aging interventions is that it takes so long to see an effect. So in kind of your tests, they really suggest that this might be surrogate endpoints for clinical trials. What data would you want to see to convince you that you can run clinical trials against organ-specific aging? And how close are we to kind of a validated endpoint that the FDA would accept as an endpoint of a clinical trial?
Yeah, that's a fantastic question. In fact, I think the aging community has realized that and put efforts into finding biomarkers of aging. So there's an X Prize that has called for basically estimating an aging intervention. It can be anything from exercise to omega-3 fatty acids to GLP-1 inhibitors or something like that, and then using any number of measurements to try to prove that you have a functional benefit, or to use functional readouts and then pair, for example, plasma proteomics, epigenetic clocks, or whatever, to show that you can basically validate the functional benefits with your marker. And I think we need to do this a few times, as you need to do in clinical medicine, to convince regulatory authorities that this can function as a surrogate endpoint. But as I just mentioned before, there are actually hundreds of really good clinical studies out there that show, for example, exercise benefits this, exercise benefits that. Taking vitamin D is good for this. And they're clinically controlled, placebo-controlled trials. There's samples there. Just need money. If somebody has money, I'd be happy to apply it to this question and really just measure, what we can measure now and see, do they correlate with the functional outcome? I think we will see this in the next 5 years. Yeah.
I have Nikolaus Kral next. Is that true still? Or you're just moving your...
Thank you so much for this really exciting lecture. In a very similar vein to what Christoph just asked, how do you translate this into the clinic? I mean, endpoints is one problem. The other one is like, this is a super highly dimensional combinatorial problem. Which proteins are causative? Which ones are just correlated? Is it a single factor? Is it a combination? What's your vision for tackling this complexity?
Yeah, and I can add, because the answer is not a single factor. How many?
Yeah.
Will there be a cocktail of 20, a cocktail of 30, a cocktail of 5? Thank you.
Yeah, no, it's a great question. Cause or effect, I think, doesn't matter as much. You know, cholesterol, cause, effect, you know, you can debate. I think many, you know, as long as it reads out with the function and is a good correlate of that, I think it might be okay. As you saw with some of our models, we only use a few proteins. So if you have 5 or 10 proteins, most of the models have sufficient power to make that call. And I think, you know, we're just starting in this field, coming back to, you know, which protein isoform do you measure, which proteoform, maybe a post-translational one, will be better than another one. We could potentially reduce this even further. But it's not very difficult now to measure protein reliably, 10 proteins. Yeah.
This is a question for drinks after, because how do you design an intervention takes a little bit more than a few minutes, but it's a good question. I have Manuel Bustinger, my Swiss mentor.
All right, my question is maybe difficult to answer, but I mean, essentially you show that the proteins are very good predictors to actually tell you what age has this or that organ. But the big question is, what is the molecular cause for rejuvenation or aging? Do you really, are the agents in the blood or are the predictors in the blood? And that if you want to study what rejuvenation and aging really all is about, molecularly, you have to go to the organs. You have to go to the immune system to exactly ask how is the composition of the immune cells changing, and it's not the blood that will ever give you a clue as to what the molecular key is for this. Or maybe you tell me otherwise. Yeah. You can at the end.
Tell him that PAX6 is in the blood.
Jolie, I should know it's not PAX6.
Okay. You feel like you can answer?
Should I answer it?
No, you can answer it.
No, obviously we do measure correlates of you know, the protein levels in the tissue, and we pick them up in the blood, but in the action is in the tissue, clearly. That's, you know, the tissue is maintained in the tissue, but it's maintained by factors that can be in the circulation. And so, you know, some of them we measure, you know, hormonal factors, you know, they obviously manage our body, and hormonal factors, many of them are proteins. So these are just, it's an extension of that concept, whether you call them endocrine factors or signaling proteins. But what's interesting is that almost every protein ends up in the blood somehow, and we can detect it. And some of them are readout of function and others are not. Mm-hmm.
Thank you.
Okay, we have now a gentleman in green in the back. I think it's green, maybe blue.
Hello, it's green, thank you. So, thank you very much for your talk. I'm Franco Lombino from Gaia Novarino Lab at ISTA. A couple of questions from the bench, actually. So the first one is, in the mice that you basically rejuvenated, right? So you, I think, performed some behavioral tasks. I was wondering, and you mentioned the stem cells of the brain. So do you attribute these rejuvenation to the activation of the neurogenic niche, or do you have any other structural or molecular evidence that this is happening? And I ask these questions because, of course, these neurogenic niche at the adult stage are still controversial, so it could be nice to have some more indications of this. And maybe for the second, I can just briefly mention or ask if those proteins that you see from the brain are directly an indication of brain function, are also these cell agers, right? Or they could also be a sign of impairment of the blood-brain barrier, for example. Thank you.
Short answers.
Okay. Neurogenesis is controversial in human brains. I think in rodent brains it's very well established. We cannot exclude alternative targets. We see an overall reduction of inflammation also. So there could be multiple, it's probably a composite of targets. And we know this also by unbiased RNA sequencing, that many different cell types in the brain respond. Your second question was, blood-brain barrier. Yeah, I'm sure that could be part of it. But it's, again, it's surprising that you see all these proteins, that you see transcription factors, kinases, cytosolic proteins in the blood. But I think it's now accepted that cells can communicate through exosomes. And many of these proteins are probably in exosomes. And, we measure all the proteins that are in the blood. So.
But your mice now, for the other way around, your mice now labeled in the brain are fantastic to study what actually increases or decreases. Yeah. I have two questions. One is, I'm sorry if I'm binary tonight, a lady in white, and then there is a hidden gentleman in the real back in the same axis.
There's also one here. Yeah.
There's this question here. Yeah. Okay, third one.
Yeah.
All right. My name is Tatiana, and thank you very much for the talk. I wanted to ask specifically because you mentioned that there is this dramatic change in plasma proteins at around age 30, and you connected it to the original evolutionary maximum age that humans without medical intervention lived at. And then there's this next dramatic change at age 65. Considering the first connection, so evolution with an age beyond 30, do you think that the second dramatic change happens because of the accumulation of the changes after the age 30, or what else do you believe is underlying that dramatic change? Thank you.
Yeah, great question. We really don't have answers for that yet. So we're trying to model this in mice now. We have 1,000 mice across lifespan, across 9 different genetic backgrounds. And I think there we can collect much more information on all the different tissues, but also in the plasma, to really start to then also manipulate these. But you see similar waves at all levels, whether you look at microbiome, metabolites, epigenetic changes. They seem to come sort of not in linear ways, but in wave forms. Yeah.
Last but one question.
Thanks for the wonderful talk. I was wondering about this age score, for instance, for heart or for brain. Let's say you have a group of people that's 40 with an age score of 60, a group of 60-year-olds with 60, and a group of 80-year-olds with 60. Say it's the brain. Do all of them have the same incidence in the next 10 years of brain disease, or is that an oversimplification of this sort of gap that you calculate?
Yeah, so we compare it only to the respective population with the same chronological age, right? So you can't necessarily say an 80-year-old who would have a 10-year younger brain is the same as a 70-year-old with a 10-year, or a 60-year-old with a 10-year older brain. Because you compare this age gap to the people who are, age 70, and then you say they're a little bit older or a little bit younger than 70. At age 80, you do the same thing. But again, it's too early to make these comparisons, and we need more data and longitudinal data as well. Yeah.
Last question here.
Thank you for the last question. A bit of maybe anthropological perspective of a question. So you sampled a lot of humans, and do you see any clustering in terms of nations or ethnical background? Because it's like we all kind of know, but how scientific is this knowledge?
Yeah, that's a really great point, and that is one of the other frontiers that we want to go. So, these studies jokingly said they're Brits, they drink tea, and unfortunately, the majority are Caucasians or Anglo-Saxons in that study, because it's a UK study. But I'm pretty convinced that we will find signatures of everything: what you eat, where you come from, what your genetic background, what your ancestry is, but also your lifestyle, everything. It's just a question of what do we look for, what we measure, what type of molecules we measure, and how transient are they? Because we know some of these proteins, they change during the day, so there's rhythmicity. If you take the same sample at different times of day, some of these proteins cannot be used. So it's just to say, yes, there is a lot of information that can still be unlocked.
Okay, so for the sake of time, we stop here, but we have a reception downstairs, so all of you who still want to ask questions, first thing, he needs to get a drink downstairs and breathe, but then you're allowed to ask him more questions and so. I would like to also thank all the microphone bringers and certainly the projectionists for all of this, and certainly before I call on stage the musicians, I would like to this to you.
Oh, thank you so much.
And this to you, dear Christina, with all big thank you from all of us. Thank you very much.
Thank you.
Fantastic. Fantastic. Jesus Quartet. And now, let's all go downstairs for the reception. Thank you very much.